It's my privilege to introduce Jonathan Fletcher, who's actually really organized the conference. Personally, he's a physician and a scientist, and he's stellar. I benefit from his quick responses to my many questions by email and his vast knowledge. He's really helped bring many agents that we're using in the lab, that he's using in the lab, for us to use in the clinic. So without further ado, Jonathan Fletcher.
So it's a spectacular occasion for us because we love to share some of the progress, some of the sense of opportunity. Sometimes they're challenges, but also a chance to acknowledge how much of a difference the GIST Cancer Research Fund specifically makes for the GIST research here at Harvard and many other institutions. It's been a remarkable commitment that over a period of years has led to very definable difference in the work that we've done and the clinical translation. So we want to thank Tanya. I don't see Robert at the moment and the rest of your group, but all the GIST Cancer Research Fund, and I'm sure we'll come back to that. Last year, I just put a few extra slides in because, there we go, because I showed something like this last year, and then the question that you asked about benign GIST. I just wanted to come back to this and remind the group, as Chan did, that GISTs are incredibly common tumors. About a third of us who don't walk around with a diagnosis of GIST nonetheless have a KIT or PDGF or a mutant GIST. And the only difference is, so they're common tumors, the only difference is that they're really small ones. They're like 1/4 of an inch in diameter. So a third of the population has these KIT mutant GIST, bona fide GISTs. We all know that the frequency of the clinical problem is rare, and therefore, where's the disconnect? And you know, this is something we have to consider when somebody says, is there such a thing as a benign GIST? Because I would say, well, yeah, I think so. I don't want to get into trouble with this. If we've got a third of us with GISTs that never go anywhere, I think it's fair to say that we do have benign GISTs. And it used to be that for a while, pathologists like Adrian would look under the microscope and say, gee, it really looks benign. And then 1 out of every 1,000 times, the diagnostician would get burned because the patient would then turn out to have something like a GIST that progressed, that didn't have benign behavior. So not surprisingly, we've all become quite conservative about saying it's benign. And in fact, some have gone so far as to say there's no such thing as a benign GIST. They're all malignant. Now, I think that's the pendulum swinging too far in the other direction. If statistically we have a group that almost never progresses. So here's one of these little baby ones, like 1/4 of an inch in diameter. And I just want to point out, the purple things, it'll be hard for you to see in the back, these are all nuclei of cells. This is a healthy little teeny baby GIST that's never going to go anywhere. It's never going to grow up to be a clinical problem. And here's the common sequela. This is what has become of one of those tiny little baby GISTs. Can you see that there's almost none of those little purple things in there? It all looks pink. It's all dead. You know, it got to a point and it just stopped and it started to fall apart, even though it had the KIT mutation. So one of the things that GCRF allows us to do by saying, you're the experts and you're working with other experts, attack the areas of biology where you see there's the most opportunity. One of the things they allowed us to do over the past 2 years was to understand, why don't these just keep growing? What's different about them and the bad GISTs? And to use that insight to do better with the bad ones, you know, sort of the other side of the coin. Because then we can say, they start the same, but what's different about them? Also, you might, you know, this would make sense. If you're studying a bad clinical problem that started like this, then to have that thing, previous slide, if I can go back, to have that when it was at its healthy point as a reference point, to be able to say that was the starting point, that's the core basis of the GIST, that should capture all the things that are fundamental to the GIST that are really important, like the KIT mutation, that we want to be able to treat. So as we're studying the bad clinical GISTs and we find something new, it's very useful for us to be able to go back and say, was it already there when it was a baby? Because if it was already there in that little small GIST, it makes a better target. It's probably more important, okay? We could talk about that more, but now the rest of the talk, that was just to follow up and to point out that this issue of benign, because you really have these two very different kinds of GIST, the very common, very benign ones, and then the ones that are diagnosed in a person who has a mass that you can see in an X-ray, and you ask, can some of those be benign? The conversation gets a little bit more complex, and when those questions come up with benign, I think you need to, again, put the expert heads together and decide for that patient, what do people think, rather than trying to generalize. Okay, now here's the thing I think is cool. So this is what Just Cancer Research Fund is allowing us to tackle that no other group I think would easily provide us the funding to do. That I'm going to go out on a ledge and predict that in 2 or 3 years, this is really going to turn out to be something. Okay, right now it's embryonic, although the evidence is very strong. The question I'm going to start with for the group is, if you have GIST which has spread in a way that it can't be cured by surgery, what Dr. Rout does, it's metastatic, and now you begin imatinib therapy, Gleevec, what is the biggest problem that you face? I'm asking an obvious question, so don't scratch your heads too much. What is the problem that the GIST population faces? Metastatic GIST, on imatinib, controlled for a time. What is the problem? Toxicity, okay. But I'm talking, and that could be life-threatening, but now I'm pushing this in the direction of what is the life-threatening eventuality, and why? Mutation. Resistance mutations, okay? And so what several of the previous speakers have already covered, if you start with a KIT mutant GIST, as most of them are, the hot spot for the mutations is this exon 11. It hits the protein right here. That's the cell membrane. Part of the KIT is inside the cell. This part is outside the cell. KIT exon 11 is the biggie, okay? That's the primary mutation that started the GIST from the original normal cell. And we know that one of the big problems is resistance mutations, where you get a second mutation on exactly the same DNA strand that has the exon 11 mutation. So that's the primary mutation. Now we have a second mutation, and it changes the structure of the KIT protein so that the drug Gleevec can no longer inactivate the target, can no longer shut it down. That's drug resistance, right? So that's the problem that we, of course, worry about so much. The primary mutations, typically exon 11 and 9, and the resistance mutations are usually exons 13 and 17. And let me just, I can't back up. Okay, that's okay. This is a patient who had 4 sites of metastatic disease, 2 in the liver, 2 in the omentum, the fat pad that covers the intestines. And they were controlled for 1.5 years on imatinib therapy, on Gleevec. But then at that point, these progressed. They all have the same primary mutation, but the second mutations, the resistance mutations, are different. And we know that depending on what kind of second mutation you have in each progressing metastasis, some of them will respond to second-line sunitinib or sutent. This one responds, but other ones will progress. So you can go from having a terrific response to Gleevec, then you start sutent, and some of the progressing disease responds and some of it doesn't. We have a problem of heterogeneity of resistance. So we have to do something about that. And here's where I think the remarkable opportunity is. So if we could accept, we could argue this later, but if we could accept that many of us would feel that resistance, that second mutation, is the life-threatening challenge that one faces after starting Gleevec for metastatic GIST. The question is, I would submit to you, can we take this second mutation, which changes the structure of KIT, which activates it in abnormal ways, and turn it into a vulnerability for the GIST cells, okay? And here's the evidence for that, okay? So this is the way KIT or PDGFRA, if you have that kind of GIST, sends its messages through the cell, different so-called signaling pathways that tell the cell to grow or tell it to survive. Some of them tell it to invade. And some of these pathways we've mapped out pretty well, so we're getting better at defining attack points. We might come back to this at the end of the afternoon. The attack points in these pathways that you can use as additional therapeutic targets, okay? So Darwin. we all know survival of the fittest, right? Those who have a gene pool that allows them to do well in a certain environment. But the flip side of the coin to survival of the fittest is fitness, that if you change the genetic structure a little bit, you change the fitness of the cell or the organism, the person in this case, or a tumor. So some of the change may be good, and some of it may be bad. What's the good thing about the resistance mutation for a GIST cell? Well, of course, it enables those cells to grow in the presence of Gleevec. It's a lifesaver for the cells. What's the bad thing? That's the part that we want to define. We're assuming, based on the evidence, that there is some liability, there's some stress to the cell that goes along with that second mutation. We have to figure out what that is and turn it to our advantage, because of all the cells in the body, which are the ones that have these primary and the second mutation? Only the GIST cells. If you can figure out what the stress is that the second mutation creates in the GIST cell, there has to be some, that's a fact, then you can define a therapy which is selective for the GIST cells that has no impact on the normal cells which don't have that mutation. Okay, so here are some unexpected observations, and this is why I'm a little bit fired up about this, because none of this would have been predicted. We know that in the patient, if you have the primary mutation, now the second mutation, it's a problem, it's drug resistance, the GIST is going to grow through the Gleevec therapy. When you take those same GIST cells with the second mutation, and you can demonstrate that the second mutation actually increases the level of KIT activation. The primary mutation, I'm going to show a couple slides quickly, increases it partly. The second mutation increases it even more. But you would think the correlate for that, if activating KIT causes the cells to grow, right, is that the second mutation would make the cells grow more, right, than the first. But actually, these cells with the second mutation grow more slowly in every model that we've created compared to the original cells that had just the primary first mutation, the ones that were sensitive to Gleevec. And remarkably, there's a standard model where you take the GIST cells from a patient and you put them into mice, and you can grow them because the mice are immunocompromised. And the cells that have only the primary mutation will grow in the mice, but if they have the second mutation, they will not grow in the mice. The cells no longer work, okay? So again, keep thinking. This thing that is our enemy, that makes the cells resistant to medicine, it actually doesn't work very well in a lot of contexts. Okay, so back to Darwin. Okay, so what are we talking about here? This is, as the expression goes, the classic double-edged sword, right? One aspect of the sword, it helps the cells, and we're saying, oh, there's another aspect of the sword too, which is a stress for the cell. Now here's my fire model, okay? If you hate these, I don't blame you, because I put these together this morning while the other speakers were talking, okay? But, so KIT in a normal cell has some energy to it. It has some level of activation. You don't have to have a mutation for activation, but we're saying it's like a low-level flame in a normal cell. ICC is the normal cell counterpart for GIST. In the presence of the mutations we've been talking about, then it gets a lot more energetic. The heat increases. We have a lot more fire that activates KIT so that those pathways that it phosphorylates, that it fires up, are effective in telling the cells to grow, to survive. And now we have a tumor, we have a problem, okay? So here's the level of activation that results from a mutation, and here's the baseline. And then, of course, if we take that GIST, if it has an exon 11 mutation that's super activated, and treat with Gleevec, we don't totally shut down the activation, although we like to think we do, but we shut it back down close to the baseline level. That can no longer support the cells to grow and to survive. And what we're now proposing is that we need to take this level of abnormal activation that comes from the mutation, and we're calling this the optimal energy level from the selfish standpoint of the GIST cells, and we need to turn the heat up even a little bit more. All of our correlates have told us that this lets the cells survive in the presence of drug, but it also creates a stress. If we can activate even a little bit more, turn this into a bonfire, only in the GIST cells I'm talking about, not the normal cells in the body, then we're proposing that this would destroy the GIST cells by creating an untenable amount of stress, okay? So here's the last of the kind of dopey pictures, but this will better get at the concept I'm discussing. This is the world's first V8-powered skateboard. Here's a rocket-propelled, these are real machines that were built, a rocket-propelled Beetle, and here's a propeller, gas-propelled skateboard. These will go fast, but we would hardly recommend them as a safe, stable transportation. And the bottom line is we're saying, put a few extra mutations in the GIST cells because they need that to survive drug therapy, and we have the equivalent of a skateboard with a V8. It's an unstable, hyperactivated situation, and we shouldn't have to be rocket scientists if we work on it hard to figure out how to turn that into a huge liability for the GIST cells. I think it's the coolest concept ever because we're saying we're taking the thorn in our sides, resistance, and figuring out how to make that a death blow for the GIST cells. We're turning it around back at them. Okay. I've taken too much time, so I'm just going to go into the conclusions and summarize what I've said. KIT and PDGFRA imatinib resistance mutations, although we visualize them as the enemy, and they've ended too many lives at this point, they nonetheless slow the GIST cell growth compared to the cells that weren't resistant. They don't actually speed it up in all of the experimental models, including mouse models, and therefore we are not just proposing but expecting that with the right approach, these resistance mutations can be exploited as GIST vulnerabilities. I'll just give you one scenario to end. We have KIT at the top of the chain, which is now hyperactivated by the second mutation. It's too activated. And we have these pathways that are now hyperactivated as a result. The cell is struggling. If we come in and activate one of those pathways a little bit more, those downstream pathways, not block it, actually increase the activation, then that could be the straw that breaks the camel's back. We usually think about, and we should continue to do so, about inhibiting crucial pathways, but this is the flip side of the coin where we say too little is not good for the cells. We know that. Too little KIT, not good for the cells. Too much is also not great for the cells. There's this concept in biology of just right. You need the just right level. So we don't just have the opportunity of inhibiting, we have the opportunity, if we do it the intelligent way, of activating. So I'll stop there. Yes? No. Researchers are struggling with immunotherapies in the sense of an antibody against KIT, which ends up for various, we might come back to that at the end of the afternoon. It comes back, ends up being a little more complicated than we might have thought. For those of you who know about the model of Herceptin, for instance, an immunotherapy against HER2 in breast cancer, we think the challenge of immunotherapies targeting KIT and PDGFRA is a little bit more complex. But then when you move on to even more nuanced and elegant approaches, like using the immune system to buttress an attack upon KIT, a conventional attack, or using these as surveillance markers, the field hasn't moved there yet. No. Okay.